Welcome everyone to week number 5 in Bio 120. We are past the halfway point now in the term, so that is good news. You should be getting an email message from me during this week after I have graded all week 4's assignments, so be watching out for that. With that being said, let's see what we've got going on. So last week you learned about metabolism. Metabolism is all the biochemistry that happens in cells, whether you're breaking down food to give you energy or you're building up and making your own macromolecules in order to be the organism that you are. That's metabolism. Then there's photosynthesis that we learned about, and that's the fact that the autotrophs in the world, auto means self, self-feeder is autotroph. The self-feeders make their own food through the process of photosynthesis. They make glucose, and most of them make glucose through sunlight energy. There are a few bacteria that use other energy sources besides sunlight, but most of them on the planet that are autotrophs use sunlight. And so we studied about that. We learned how they're related to each other. We learned the importance of ATP. All of that is important. All right, so this week is all about DNA. It's one of my favorite weeks. What was my dissertation on? DNA fingerprinting, DNA profiling. I'm just saying, so it's one of my favorite topics. Skim through chapters 10, 14, and 15. They're all about DNA. I say skim because these chapters are very heavy on jargon that's used in genetics. Genetics has its own language above and beyond what we use in the rest of biology. So I'm just saying there's a lot of terminology in there, and you don't have to memorize what's in these chapters word for word for word. One important feature of this OpenStax textbook is that you can search it. So I suggest that you do that when you find a question that the assignment is asking you, like mitosis. Just type in the term mitosis in the search bar and it will take you to the sections of the chapters that discuss mitosis. So use that search feature in your online textbook. That's an advantage to that online version. All right, I want to say a couple of things about DNA before we move forward. One thing is that DNA is the molecule of heredity. It contains genes. Genes are little segments of the DNA that are used to produce proteins. The proteins, most of the proteins in cells are enzymes. Enzymes have particular jobs. They carry out a chemical reaction of some sort. They have very, very specific jobs. They do one thing and one thing only. And so it takes lots and lots of enzymes to carry out any biochemical pathway, like photosynthesis. There's so many enzymes involved in photosynthesis, and each one is specific and has a specific job. So it's really important that your DNA stay nice and pristine so that you get all these specific enzymes that you need to run your biochemistry. DNA is replicated before the cell makes a copy of itself. Replicated just means it makes a copy of itself. That's all it is, replication. Make a copy. And so it has to be done very well and very exactly. And it does happen like that. But once in a while, a mutation occurs. Are all mutations bad? No. Most of the time, a mutation, which is just a change in the base sequence of DNA, most of the time a mutation doesn't cause any issues. Sometimes it causes problems. If it's in a really, really important gene for a really, really important enzyme, yes, it can be detrimental, even deadly. Sometimes the mutation is a good one because it produces a new enzyme that gives you some new trait. So mutations are not always bad. All right, so that's DNA and replication. And the other thing that I want to say is that the job of DNA is to make all these enzymes. The job of DNA is not to make a copy of itself. That's just so that you can get a new cell and move on into the future with new cells, right? But the job of DNA is to make all these enzymes. So don't forget that this week. All right. I'm going to talk about the cell cycle here in just a second. Your discussion board postings this week, remember your initial post is due by Thursday. Your 2 response posts are due by Sunday. For your initial post, you're going to be discussing cancer with one of your friends. And one of your friends knows a little bit of biology. And the discussion is cancer a problem with cell division, or is it a problem with genes? A problem with cell division means, is it a problem with mitosis, where the DNA is replicated? Remember we talked about that. When the DNA is replicated. Or is it a problem with the genes? We just talked about that too, where genes are mutated. Or is it both? Remember I told you last week? If you ever see a question like this, you know the answer is both, or else they wouldn't have said or both. Yeah, just saying. So the cell division thing, I haven't talked about that, and I want to mention this over here with the diagram that I put here. There's a video on the cell cycle. There's a diagram in your book. Okay, I like mine better. I'm just saying. So I'm going to explain it to you. So the cell cycle is the life cycle of a cell. That's all it is. It starts, and then there's cell division up there at the top where you get 2 new cells. Okay? So right at the 12 o'clock position, that's where the life of the cell starts. All right. So going toward the left. Oh, sorry. Oh, geez. The right. Going toward the right. Like you do on a clock, right? You have G1 phase. G1, G used to stand for gap because we didn't know what happened. Okay, now we know what happened. So I don't know why we still call it G. But anyway, it's the growth phase of the cell where the enzymes increase in number, where mitochondria increase, so you have enough mitochondria to produce all the energy that you need in cells. There's all kinds of basic cell growth happening in the G1 phase of the cell cycle. I'm coming back to G0 in just a second, okay? So G1 starts at 12 o'clock and goes all the way down to about, I don't know, 4 o'clock there. And then we have the S phase of the cell cycle. S stands for synthesis. This is where the new DNA is produced because the cell is starting to get to the place where it's going to divide. So it makes its new DNA in the S cycle, the S phase of the cell cycle. Then you have G2, the G2 phase. This is where all the cell machinery that's needed for cell division is produced. The centrioles, the spindle fibers. It depends on the type of cell, and I'm listing some that happen that are produced in eukaryotic cells like our cells. That's G2, the general cell machinery that's needed for cell division is produced during G2. And then you move over to M and C, and M stands for mitosis, and this is where you have the splitting of the chromosomes into the 2 new daughter cells, because you've made new DNA in that S phase. And then C stands for cytokinesis, and this is where the rest of the cell splits apart and makes 2 new daughter cells. So at 12 o'clock then, when you get back there, you have 2 daughter cells, and each one starts back into the cell cycle. Now, the purpose of a cell is not to go through the cell cycle. This is just explaining how cells divide. The purpose of a cell is to do some work. Well, where does that work happen? That happens in G0. So cells stop. They stop the cell cycle in G0 so they can do their work. This is where they're mature enough, they're big enough, they have enough stuff inside their cells to do their work. They stop here and they do their work. That's G0. One problem with cancer cells is that they never stop in G0. They just keep dividing and dividing and dividing and dividing. And yes, there are lots of mutations in cancer cells, and so you have all kinds of strange, crazy things going on in cancer cells because of this cell division that is continually taking place. And the work of that particular cell is not being done anymore. The whole purpose of a cancer cell is just to divide and divide and divide and take up more and more and more of your nutrients. So that's mitosis, the part of this. And genes already talked about with mutations, right? So a mutation is a change in the genes, in the DNA, and that changes the function of these genes. And in this write-up, it talks about tumor suppressor cells and proto-oncogenes. You should look those up because they're very important in cancer. And then you're going to also answer the question, how would lifestyle changes lower your cancer risk? Everybody knows that you shouldn't smoke, right? Everybody on the planet knows this. That's one way to lower your cancer risk. Are there other things that you can do? Sure there is. Avoid air pollution. That's another one. There's so many others. So discuss that. And then in your response post, you're going to talk about environmental carcinogens. So a carcinogen is anything that causes cancer. That's what it means. Usually we're talking about chemicals, but there are viruses that are carcinogens. Okay, so anyway, environmental carcinogens means carcinogens that are out there in the environment that you're exposed to. So you're going to discuss these and what would happen and how beneficial that would be if we remove them from the environment altogether. So those are your response posts. All right. So those are the discussion board postings this week. You also have milestone 3 of the final project. And this is the third part of your final project. Then after this, you're going to start putting all this together. So I'm going to quickly go through these steps and what you need to discuss in milestone 3. But before I do that, I want to tell you that the format this week is totally different from the other milestones. Look down at the bottom. You're required to make PowerPoint slides this time, 6 to 10 slides, and you're going to outline this information on the slides. All right? And of course, all of these are going to have these headings. You're going to have a slide that says milestone 3 of the final project. And then each slide, you can count how many sections you have here. That's how many slides you should have. You're going to outline these topics. So what are the methods of conservation that would help prevent this particular issue that you've been investigating? How could we prevent global warming? We could stop burning fossil fuels. Duh. I mean, that's pretty easy. But there are other things that we could do. So how could we prevent this? That's conservation. How do we stop this from happening in the first place? Education. Why is education important about this topic? Why do people need to be educated about global warming or eutrophication or plastic pollution or anything else that you have as a topic? What are some solutions to this? So how do we fix it? How do we fix the amount of carbon dioxide that's already in the atmosphere that is too much? It's too much there. It's trapping heat. We could plant a bunch more trees. Yeah. There's a movement called Billion Tree Movement, I think it's called, where we're trying to get more and more and more trees built instead of mowing down every single one when a new subdivision is built. Yeah, it's one of my pet peeves. Ethical considerations. Are there ethical considerations about this issue? This kind of goes along with the next part too, positive and negative aspects of the issue. There's got to be some benefit to somebody out there in the world to burn fossil fuels. I think we can figure out who, right? The big oil companies? Yeah. And people who own stock in those big oil companies? Yeah. So anyway, you can figure this out. That's an ethical consideration. Should those people be allowed to do this and make all these profits off burning fossil fuels when we're totally destroying the planet? Probably not. So that's an ethical consideration. Positive and negative aspects of the issue. There's got to be something that benefits somebody somehow. when we're talking about plastic pollution, right? Why did we switch over to everything being packaged in plastic? There has to be some reason that we did this. So investigate that. And what are the negative aspects of the issue? And then personal impact. How can individuals change their behavior, change what they're doing in order to help fix this problem? So personal impact. Okay, so that's your milestone 3. Include citations and references. Citations are you referring to your sources in the body of your work, where your references are at the end. And label all your sections. This is very, very important. Okay, right here, you can stop the video if you want to, because the next slide is me explaining DNA fingerprinting, also called profiling, also called DNA typing. Oh, it goes by so many names now. But since that is one of my special areas of interest, I'm going to explain that to you. But if you want to stop here, it's perfectly fine. Remember that if you need anything, m.sigmon@snhu.edu. All you have to do is ask me questions. I'll be glad to answer anything. If you're confused at all, especially with the sections this week, you may have some confusion. So just let me know, please. And with that being said, I'm going to move over to DNA fingerprinting, and I'm going to get my face out of the way. There you go. So this used to be called DNA fingerprinting back in the 1980s when Sir Alec Jeffreys, he was not knighted at that time. Here's a picture of him right here. He was a geneticist in England, and he developed this technique where he could chop up DNA and then look at parts of it that were variable between people, right? There were differences in the DNA in these little sections between people. He figured out how to do this particular technique. And over on the right-hand side over there, you see a picture of an x-ray of some DNA fingerprinting from the original technique. And this is probably what you recognize from something that you've seen before. And they call it barcodes, right? It looks like barcodes on food products in the grocery store, that sort of thing. So in 1985, Alec Jeffreys had published a paper on this technique, and he had done studies of, oh man, I forgot the group. Can't remember if it's Mormons or, but it was a closed sort of society, and he looked at the relationships between the people to see if he could determine differences between the individuals using his technique. And he could also figure out relationships between them. And he said, yes, this actually works. So he published this paper in a scientific journal, and some police officers in England had heard of this. And they said, I wonder if this could be used to help us solve crimes. Well, they had a particularly disturbing case. There had been 3 teenage girls who were murdered in England in 3 different villages in the same way. And so they suspected that they had someone who was the killer of these same 3 girls. And back then you didn't even have the term serial killer, but now we do. They thought there was a serial killer loose. But the only evidence they had was semen that was left at the scene of the crime. And they didn't have anything else, no eyewitnesses, no nothing else. So they went to Alec Jeffreys and they said, do you think that we could test this DNA that we have against men from the local area and find the perpetrator of these crimes? And he said, yeah, I think it should work. And he said, but what you're going to have to do is you're going to have to collect blood. That's back when you had to do blood. You couldn't even do a buccal swab, which is a cheek swab like you can now. He said, you have to collect blood from all of these men. And they said, we can do that. And so people in England don't have the same civil rights as we do. And so they just required men to come in to get blood samples. So they did. And Alec Jeffreys started testing all these blood samples, and he was going through hundreds and hundreds and hundreds of them, finding no match, no match, no match, no match. And, you know, everybody was getting worried. Well, if you're the criminal and they're asking people to come in and give blood, aren't you going to figure out some way to not do this? Well, this one fellow named Colin Pitchfork, honest to goodness, that is his name. Colin Pitchfork asked his friend Ian Kelly if Ian Kelly would go in and give a sample of blood, his second sample, to the police and pretend to be him. Ian Kelly said, yeah, sure, whatever. So he did. Well, later on at the pub, Ian Kelly was hanging out with a group of his friends, and he was getting a little bit tipsy, and he told everybody that he had gone in and given blood a second time for Colin Pitchfork. Well, one of the women in the group said, that's a little suspect. Why would anybody do that? And so she went to the police and told the police, and they went and grabbed up Colin Pitchfork, took some blood from him, figured out that he actually was the perpetrator in this case. He had killed these 3 girls. He could absolutely have been not a suspect on anybody's radar for any reason. He had a steady job. He had a good home life. He had a wife and children. Never had been in any trouble whatsoever. No one suspected him whatsoever. And since then, he has been convicted of the 3 murders, and he is still in jail in England. Well, of course, this made headlines all around the world. Alec Jeffries began calling this DNA fingerprinting. Actually, it was his wife, because he described to her what he was doing, and she said, you know what, this sounds like fingerprinting, doesn't it? Because with fingerprints, you can distinguish between people. He said, yeah, so let's call it DNA fingerprinting. Well, it doesn't have anything to do with fingerprints. So now it's called DNA profiling, DNA typing. There's all kinds of terminology. This particular technique is called the RFLP technique, restriction fragment length polymorphism, which means nothing to you, but it gives the barcode, like over there on the right. Then later on, we discovered a new, faster technique that required less DNA and less pristine DNA, and that's called PCR, the polymerase chain reaction technique. This is the technique that's being used now, and it's even more sophisticated now than it was in the 1990s and the 2000s, because we can look at single differences in the DNA now. They're called SNPs, single nucleotide polymorphisms. We can look at single differences in the DNA to distinguish between people now with the newest techniques. So it's come a long way since 1985. We've been using it in forensic science since then. In 1988, the FBI started using this, which was then followed by different states setting up their own laboratories. So this has been going on since the 1980s. Some of the latest things that have been happening, genealogy websites have been used, sometimes without anyone's knowledge or approval, to search for criminals in their databases. This is called forensic genealogy. The Golden State Killer, Joseph DeAngelo, was discovered this way over 40 years after he had killed numerous people and committed all kinds of other atrocities in California. But also now we're finding out identities of missing people this way, bodies that were discovered decades and decades ago, and we don't know who they are, and we're determining their identities through websites like this. So we're using this technology to help us gain information in forensics and missing people, family ties, all that sort of thing. I'm sure you've heard of it. It's very interesting, and it's becoming more and more precise as we go. Every year, something new and different is happening. And so I just wanted to share that with you. And if you want to know more, all you have to do is ask me, because I love it. Be glad to discuss it with you through email or however. So anyway, that's just a little background on DNA fingerprinting, also called profiling and typing, and also the new science of genetic genealogy. With that being said, I hope that you have a wonderful week this week, that you're doing well, that you're not sick, or that you're not sick anymore. And that if you need anything, please just let me know. m.sigmon@snhu.edu. Have a great week.